All right, we ready? Not too bad so far, right? I hope. Okay, so today's couple lectures we got are going to be a little more dense than yesterday, so I'm going to go at a little bit faster pace. Hopefully, most of you, if not all of you, have seen this at some level before because it's on the MCAT. So even if you haven't taken a formal class, I'm sure you've had to at least look at this stuff before. So today we're going to talk about, the title of the lecture is Nucleic Acid Chemistry and Chromatin. That wasn't my choice for a title. I think we'll change it in the future. It's just nucleic acid structure is really what we're going to talk about. So it sounds more confusing than it really is. And there's the learning objectives for you guys to use as you see fit. So the first thing I'm going to mention, kind of keep things simple at first, central dogma of molecular biology. This is going to kind of outline what we're going to do over the next 4 to 6 lectures. I forget exactly where the cutoff is. But DNA, and today we're going to talk about the structure of DNA and the structure of RNA. But DNA stores genetic information, right? Gets replicated, gets transcribed into RNA, and that RNA gets translated into protein. That's a central dogma of molecular biology. Okay, and that kind of outlines a lot of what we're going to do. So in order to discuss DNA replication, transcription, translation, the first thing we got to do is look at the structure of these nucleic acids. So the first thing we're going to look at is the building blocks, which are going to be our nucleotides of DNA and RNA. So the first component of a nucleotide is a 5-carbon sugar. So there is a sugar inside your DNA molecule. Most sugars you think about in the body, like glucose, fructose, those things, are actually 6-carbon sugars. So later on, I'm going to show you the pathway where we actually derive the 5-carbon sugar from. But anyway, in RNA we have ribose, and in DNA we have deoxyribose. So really the only difference between these two is on the deoxyribose, you're missing an oxygen group. It's been removed. That actually ends up having profound effects on how that molecule interacts and its stability and things like that. So RNA is much less stable than DNA. So we have our 5-carbon sugar. The next part of a nucleotide is a nitrogenous base, okay, and that's what we've shown here. Once again, you don't have to try to, we're not going to ask you to draw out the structures, for instance. I do think it's useful for you guys to remember which ones have a single ring and which ones have 2 rings. So the purines have 2 rings. The reason being, later on when we talk about how these things are synthesized, the purines are a little more complicated, as you can see from the picture. And so we actually will salvage our purines where we don't really salvage the pyrimidines. And I'll explain later on what I mean by salvaging. But anyway, it's helpful to know that. And it's also, and one of the things I do want you to know is which nitrogenous bases are purines and which ones are pyrimidines. So the purines are adenine and guanine, okay, and these are the ones with the 2 rings. And then our pyrimidines are cytosine, uracil, and thymine. So thymine is going to be in DNA, and uracil is going to be in RNA. And so there's a little mnemonic there for you to help you remember which ones are which, because I would ask you to know that. So pure, as gold, purines, AG. So I've also seen other people use, once you know one, then you automatically know the other, so you can just use one, whatever one you like. So you got CUT here, so cut the pie. Sometimes people use that to try to remember. But that is something you would be expected to know. And once again, DNA and RNA, they have the same nitrogenous bases with the exception that DNA has thymine and RNA has uracil. And really, if you look at the difference between uracil and thymine, there's really not that big of a difference. You just have an extra methyl group. And that's going to be important in a later slide when I talk to you about transfer RNA. And in case you didn't already know this, hopefully most of you already did, nucleotides are usually abbreviated by the first letter of the nitrogenous base in that nucleotide. So A, G, C, T, or in the case of RNA, you would have a U instead of a T. And then finally, the third component of a nucleotide is a phosphate group. So you can have 1 to 3 phosphate groups, and they're actually going to be attached to the 5' carbon on that sugar molecule. So in this case, we've just got 1 phosphate group, but it can have up to 3. Okay, so it can be a mono, di, or tri phosphate. And I've included this because somebody will always ask, why do you call it 5'? Because we're going to use this term, 5', 3'. You're going to get sick of hearing that by the end of today. And that's just the numbering system on the sugar molecule. So basically, they reserved the regular numbers to number the carbons on the nitrogenous base. So they would be labeled 1, 2, 3. And then when you get to the sugar molecule, they use the prime designation, 1', 2', 3'. So that's not a detail I'm going to test you on, but I just wanted you to understand where that came from, since you're going to see it over and over and over again. Okay, so I want you to compare a nucleoside with a nucleotide, something that sometimes throws people off. If you see the term nucleoside with nothing else there, we're talking about a structure that only has the nitrogenous base and the sugar. So it's missing the phosphate groups in this case, whereas a nucleotide has all 3. Okay? But you can also use the term, for example, nucleoside monophosphate. Okay? You wouldn't call that a nucleotide monophosphate. You would call it a nucleoside, because that tells you those 2 components are there. And then if you say monophosphate after, well, a nucleoside monophosphate is a nucleotide. But if you see the term nucleoside by itself, that means the phosphate's not there. Okay, so that's just a little bit of nomenclature. So these nucleoside di and triphosphates, when you've got 2 or 3 phosphate groups on there, those are high energy compounds. Okay, and that's the energy in there comes with these acid anhydride bonds. You know, going back to OChem, right? And so the energy associated with those bonds are what drive most of the reactions in your body. And there are other things where we can derive energy from, but most of the things come from the energy in these high energy acid anhydride bonds. Interestingly, as we go through DNA, so you'll use ATP, for instance, or GTP less often, but you still see that. You use it for energy to drive a reaction, right? And I'm sure you've seen a little bit of that. If not, we're going to see it a lot in here. One of the kind of neat things about DNA and RNA synthesis is the energy to make those molecules are in the nucleotides themselves, which then becomes the structural component. Okay, so the nucleotide actually becomes the structure of an RNA or DNA molecule. But these nucleotides are also used for other things besides nucleic acid structure, besides DNA and RNA as we know it. They exist as single nucleotides to be used for the energy for many, many, many reactions we're going to see in biochemistry. And a lot of the most important reactions that we're going to see in biochemistry will use one of these nucleotides. Okay, so we're going to look at the structure of DNA and the structure of RNA. We'll start with the structure of DNA. DNA is a polar molecule. That means it's got a distinctive beginning and end, okay, and they're different. Just like when we looked yesterday at a polypeptide, it had a specific beginning and end. DNA has beginning and end. It's got polarity. At the 5' end, it's got a free phosphate group. And at the 3' end, it's got a free hydroxyl group. Okay? And those are going to be important, as we're going to see when you go to add a nucleotide to a growing chain of nucleic acid. The nucleotides in DNA, and so right now we're just showing you a single strand of DNA. DNA is normally double-stranded, which I'll show you in a second, but the nucleotides in each single strand are actually joined by phosphodiester bonds. That's what's shown right here in red, or circled in this square right here. That's our phosphodiester bond that links two nucleotides in a molecule of DNA, also in a molecule of RNA. And then you have your bases are actually joined by a glycosidic bond. You could actually just look at the nucleotide structure to see that as well, but here is our nitrogenous base, and there's the sugar component. So that's a glycosidic bond right here where this arrow is. Right here, and right here, and right here. Those are glycosidic bonds. Okay, so nucleic acids may be written a number of different ways. So I'm trying to help you keep this straight in your head. Okay, so when you're speaking about DNA and you're looking at one of the strands, the forward direction, or the direction, as you'll see, the direction we synthesize a molecule of DNA is in 5' to 3'. So that's the forward direction. Okay, so if there is no designation made to the ends of the DNA, like if I give you a sequence that just says TACG, you can assume that all the way on the left is the 5' end and all the way on the right is the 3' end. Okay, so you can either designate this 4 nucleotide section as 5' TACG 3', like I've done there, or you can simply remove the end designations and just say TACG. Okay, both mean the same thing. Once again, if it's not designated like this, it means this. However, if you want to write the sequence backwards, the ends must be designated. Now, you're probably, if you're not familiar with some of this stuff before, you're like, well, why would we ever even want to write it backwards? I'll show you in a second a good example of where you would want to write a DNA sequence backwards, at least at first, and then you can actually flip it over. So if we take this sequence, TACG, and we write it backwards, we'll get GCAT, right? That's TACG, T. So actually, one thing I want you to get out of this slide is that this sequence right here is the same as this, and is the same as this. Okay? And if you look at that slide a little while, if you've seen this stuff before, it's probably real simple, and you're like, okay, move on. I got this. If you look at it and if you're getting confused, I'm seeing you, because once you get it, you'll be like, oh, okay. So don't spend hours struggling with this. So you should be able to recognize this sequence and this sequence are the same thing. This one's just written backwards. Because it's written backwards, we have to designate the ends, though. So 3', then the sequence, 5' tells us it's written backwards. Okay? But also, you should be able to take a sequence like this. If I give you a backward sequence, you should be able to write it. You should be able to flip it and write it like this. And all you do is write 5' down and then go, let's see, T, A, C, G. Right? Okay. So that stuff's, it's pretty simple if you've done it before. If you haven't, it might be a little more difficult. Okay, so now we're actually looking at a double-stranded DNA molecule, which is the way DNA normally exists, at least in most organisms. There's some viruses that are exceptions, but DNA is normally double-stranded. And a couple terms used to describe DNA structure. One is anti-parallel. So we've got a double-stranded DNA molecule here. I talked about we have these end designations, 5' and 3'. Well, the ends of these two DNA molecules go in opposite directions. That's what we mean by anti-parallel. They're parallel, but they're going in opposite directions. So one strand is going 5' to 3', and then this strand over here is 3' to 5'. Okay? You see what I'm talking about? So they're going in opposite directions. But they are parallel. They're also complementary. That means when you look at this, when you look at the sequence in here, T is always base pairing with A, C is always base pairing with G, or vice versa. ATGC, okay? So that allows us, if we have one sequence, we can actually determine the complement, okay? And actually that sort of breakthrough, realizing that that's how things base paired, was how Watson and Crick originally discovered or solved the structure of DNA many, many years ago, okay? So that means if I give you any sequence of DNA, a single-stranded sequence, you should be able to tell me what the complement is, okay? Now I've actually, instead of breaking it up into multiple slides in the interest of time, I've given the whole thing here, but what I would do on a question if I was going to ask you this is I would just give you this. Or I may even be even trickier and not even designate the end, just give you this AGG, and you would know, you would be able to write down that it's 5' to 3', right? You know that. But I might give you a single-stranded sequence like that and ask you to determine what is the complement. Usually when I do this, the entire class gets it right, so it's not that complicated. But this is also where, in this case, you would at least initially want to write the sequence in reverse, because it's much easier to write it in reverse at first. So what I would do is take this sequence that you're given, and I would start by writing 3' and 5', so I know I'm writing in reverse as I determine the complement. Then I'm going, let's see, A, okay, that base pairs with T. G, okay, that base pairs with C. And then you just go down the strand. And then when you're done, you're left with this. Now that's actually written backwards, and that could be an answer choice, or I may have all the answer choices written in the forward direction, so you can rewrite that in the forward direction, right? So you start with the 5' end. See, we got C, C, T, A, C, C, T, A. You just reverse that sequence. See what I did there? Once again, I don't want to belabor this because I think you guys have seen this before. If you're struggling with it, come see me, because I can give you a couple practices. Once you got it, it'll be easy. Okay, so we can also use that complementary base pairing to apply Chargaff's rules to determine the amount, given the amount of any single nucleotide in a DNA sample, we can determine the amount of the other 3 nucleotides. For example, if I give you the amount of A, you can determine the amount of T, G, and C. Okay, so that's a calculation I would ask you to be able to perform. And I've given you an example here of a question. A sample of DNA has 10% G. What is the percentage of T? Okay, well, the number of the nucleotides all have to add up to 100%, right? There's only the 4 nucleotides. And you also know, well, G always base pairs with C. So if we have 10% G, we have to have 10% C, right? So that's 2 of them. When you add those 2 up, you get 20%. So that means there's 80% left. Okay, well, if there's 80% left, then there has to be an equal amount of T, and there has to be an equal amount of A, then 80 divided by 2 is going to give us the amount of both T and A, which would be 40%. Okay, so that's another thing I might ask you to be able to perform is, given a single nucleotide, tell me what the percentage of the others is. Okay, the only trick that I've ever seen on a board exam where they did this was they switched it up a little, and they actually gave you a double-stranded RNA virus. So rather than T, they had U in there, but it's the exact same rules. They just substituted T for U, okay? They were trying to be tricky, but... Okay, so we've got a couple different drawings here of a DNA molecule. And what I want you to notice is the hydrogen bonds, okay? So we have T bound to A and C bound to G. Now you'll notice the TA base pairs have 2 hydrogen bonds, right? And the GC base pairs have 3 hydrogen bonds. Okay? These hydrogen bonds, by the way, are what hold these 2 DNA strands together. Okay? And you also see it in this figure. T and A, you got 2. G and C, you got 3. That's actually becomes important. Okay? In particular, when we're talking about the denaturation and the renaturation of DNA. Okay? So you can actually separate the 2 single strands of DNA. So when you do that, you're breaking, what I'm talking about right now, we're breaking the hydrogen bonds between the strands. We're not breaking the strands themselves. Those phosphodiester bonds and the single-stranded piece of DNA are still there. Glycosidic bonds are still there. So the single strands remain intact. We're just separating them. Okay, and you can do that with heat. You can do that with alkali. You can do that with certain chemicals. So right now I'm talking about in a test tube. We can also do that with enzymes or other proteins in the cell, which we're going to talk about shortly. And then what you're left with, if you completely denature it, is a single-stranded piece of DNA. Now, the easiest way to think about this, so you can use chemicals, you can use altered pH, but I think the easiest way to think about this is heat. And then very often in molecular biology, in the lab, that's what we use. Those of you who are familiar with PCR, which you'll hear about shortly, we just use heat. So you can just use heat, and you got to get up to about boiling temperature, pretty close, before you can separate the two strands of DNA, but you can do it. If you actually remove that heat or actually cool that DNA sample down, what happens is the DNA, the hydrogen bonds will actually reform, and you'll actually convert this single-stranded DNA, assuming the complements available in the solution, you will actually reform those hydrogen bonds and get back to the double-stranded DNA molecule. Okay? So the fact that we can break DNA apart and actually re-anneal it is important for replication and transcription, which we're going to be talking about over the next couple of days. It's also important for molecular biology techniques, which Dr. Traore will talk to you about later on this week. Okay, so this property of being able to separate DNA allows us to assign a value to it called the TM. So the TM is defined as the temperature required to melt 50% of the DNA in a sample. So when I say melt, that means to break 50% of the hydrogen bonds available in that sample. And this is determined, as you can see by the y-axis here. They're going to use a spectrophotometer to determine where that 50% breakage occurs. Now, so single-stranded DNA and double-stranded DNA read a little bit different in the spectrophotometer, so that's how you can determine that. That part is just FYI. What I want you to be able to do is actually interpret these graphs, okay? So, because heat, in this case, is actually breaking the hydrogen bonds, if you have regions of DNA which are particularly rich in AT base pairs, which have fewer hydrogen bonds, those will actually separate into single strands at a lower temperature versus a region of DNA that has a lot of G and C, which has 3 hydrogen bonds per base pair. Okay? So what that means is if you have a DNA sample with a high GC content, it's going to have a higher TM. Or conversely, if you have a DNA sample that's very high in AT base pairs, it's going to have a lower TM. So when you're looking at this graph, and we're going to determine the TM, that's for half of it. So when we get way up here, you have DNA that's fully melted, 2 single strands. And way down here, you're going to have DNA that's fully double-stranded. As we apply heat, it's going to start separating, and that's what's going on. As the temperature goes up, it's separating. At some point, it's about 50% separated, and that's our TM. So when you look at these 2 graphs, and now it's actually labeled, so it tells you there, but hopefully if this is not labeled, you could look at that graph and you could say, okay, this DNA sample has more GC in it, for sure, because it's melting at a much higher temperature. This one's actually pretty dramatic. You see that? Not too bad, right? So I may ask you, for example, to interpret a graph where I give you TM values. Okay, so now we're going to look at how the DNA is actually organized. So one thing to keep in mind is that the genomes of most organisms are huge compared to the size of the cell. So for example, in a human cell, if you stretch the DNA out, it's about 2 meters long.
And a typical human cell is, I mean, it varies depending on what cell type you're talking about, but let's say 10, 20 micrometers. So that's a huge piece of DNA if it's stretched out to try to fit not only inside the cell, in eukaryotes, actually all inside the nucleus. So somehow we have to compact that DNA. And that was just a little FYI. It's not something I expect you to memorize about the length of the DNA. It's just to kind of wrap your mind around how huge the length of the DNA molecule is. So we have to compact it somehow. In prokaryotes, and actually in mitochondria have their own DNA, and it actually looks very much like a prokaryotic plasmid. So we're also going to do this, but in prokaryotes, we actually will supercoil the circular DNA. So if you didn't know this already, prokaryotic chromosomes are circular, and eukaryotic chromosomes are linear. So the DNA in your nucleus is linear, but in bacteria, it's going to exist as a circle. So what you do if your DNA is a circle and you actually have to compact it is you supercoil it. Okay, and so basically you're taking this big round ring and you're actually twisting it back on itself. That's what supercoiling is. Okay, and that allows it to be compacted further and fit into, in this case, in a prokaryote inside the cell. They don't have a nucleus. The enzyme that actually does this, and we're going to see this enzyme again in the next lecture, is DNA gyrase, which is an example of a type 2 topoisomerase, and I'll show you what a topoisomerase is in the next lecture, so don't freak out about that yet. This particular enzyme, which actually supercoils this plasmid in DNA, is inhibited by the quinolone antibiotic. So here's our first antibiotic I'm going to introduce. I'm going to introduce several classes of antibiotics to you guys. You'll certainly expand upon those later. But whenever you see this fluoxetine at the end, it's some type of quinolone antibiotic, and it inhibits prokaryotic DNA gyrase. Therefore, it actually ends up interfering with the ability for it to supercoil its DNA. Also, in DNA replication in prokaryotes, it actually inhibits when you replicate a circular piece of DNA, you actually end up with two rings locked together, and this DNA gyrase is actually allows you to separate those two rings. So it actually prevents the bacteria from actually dividing. So it specifically affects bacteria. That's why it's an antibiotic. Now, in humans and other eukaryotes, the DNA is all contained in the nucleus, with the exception of the little bit of DNA in the mitochondria. And the DNA in the nucleus is packaged in chromatin. Okay, so chromatin is a mixture of DNA associated with histone and non-histone proteins. So chromatin itself is made up of packaging units called a nucleosome, and I'm going to show you that's what a nucleosome is right there. Okay, so histones are rich in lysine and arginine. We talked about that yesterday, right? These are basic amino acids. They have a positive charge. So I guess I probably didn't explicitly note it, but if you go back and you look at the pictures of the DNA molecule, you have that phosphate backbone all through, actually on the RNA too, but you also have it on a DNA molecule. That phosphate's negatively charged. So DNA is very negatively charged. And so histones are rich in lysine and arginine because they're going to bind to, so they're positively charged and they bind to that negatively charged DNA. So you actually have two copies of histone H2A, H2B, H3, and H4, which are actually shown in this little figure. They're going to aggregate to form this histone octamer. That's what's shown right here. That's our nucleosome. Now you have to kind of use your imagination because there's two copies of each of those proteins. So we're kind of looking down in this particular picture at this nucleosome. Underneath there's actually an additional copy of each one of those. So you have that octamer, 2 copies of H2A, H2B, H3, and H4, and then you wrap the DNA around it. That's our first level of higher order structure of DNA in the nucleus in eukaryotes. So if you have a group of these free nucleosomes, which is just shown right here in this part of the figure, without this H1, which I'm going to get to in a second, we call this the beads on a string model, okay? So you do have regions of that DNA that are a little bit exposed, and there are nucleases around. So one of the things the cell has to do, and it's kind of a central concept in biology, you have to balance between times when DNA needs to be exposed, like when you want to transcribe it or when you want to replicate it. But DNA is your hard copy of genes. You also want to protect it, okay? So at times when DNA is not being used for anything, you want to have it highly packaged up. When it's being expressed or being replicated, then you actually will want to expose it a little bit more. But you have to maintain a fine balance because that exposure also opens it up to damage, okay? So the last histone specifically that you guys would need to know is histone H1, and that's actually going to bind in between these nucleosomes. And when that binds in there, we actually causes the DNA to form this structure right here, which is the 30 nanometer filament. Okay, that's the next level of higher order structure. Okay, so here's our beads on a string right here. That's where we don't have H1. You add H1, and then you form this 30 nanometer nucleofilament. Okay, and that's as high a level of structure as you would need to know to move on and for board exams and things like that. Now the DNA does get much more condensed than that. So those nucleofilaments actually fold back on themselves. And there's actually a whole bunch more proteins involved. And you can actually condense your chromosome so much that it's actually even visible under a light microscope, which is kind of the classical chromosome pictures that people think of like during mitosis. That's as condensed as they ever get. Okay? And that's much, much more condensed than what we're seeing right here. But there's no high-yield proteins or structures we need to learn beyond this. Okay, so these are the histones that you would be expected to know. Okay, so next we're going to look at RNA structures. In many cases, RNA, if you understand DNA structure, you know how DNA is formed, you're real comfortable with that. You're 80 or 90% of the way there for RNA, okay? Maybe even 100% of the way there. You just got to cut off a couple things to think about RNA structure. In some ways, RNA structures are actually more interesting than DNA structures, but in some ways, they're more simple. It depends on your point of view. So we already saw this. DNA contains deoxyribose and RNA contains ribose. In case of the sugar is a little different. DNA contains thymine and RNA contains uracil. So if you're looking for the complementary RNA, you have to remember RNA contains uracil instead of thymine, with one exception, which I'll talk about shortly. DNA is double-stranded, as we saw. A mature molecule of DNA has 2 strands under normal circumstances. However, RNA is considered single-stranded. Most RNA is single-stranded. Now, I have to always use the term most when I'm talking about DNA and RNA. For every rule that I make for you guys in molecular biology, you're going to find out that there's viruses that break that rule. Okay, so that's why I have to say most. Okay, so there are double-stranded RNA viruses, for instance, but RNA that exists in eukaryotic cells is normally single-stranded. However, RNA can base pair back on itself, which follows the same complementary rules that we learned for DNA base pairing. what RNA can do is actually RNA can fold back on itself where it has complementary regions, and we'll see that. A good example will be transfer RNAs, okay, which we'll talk about shortly. Actually, base pair, the whole thing won't be base paired, but there'll be little sections, let's say 10, 20 nucleotides that are complementary that'll base pair. So you get these like stems and loops that you'll see. Generally speaking, RNA is much smaller than DNA. Okay, most DNA exists as a chromosome. Chromosomes in humans are huge. Even chromosomes in bacteria are huge. Sometimes if you have a plasmid or you have the mitochondrial DNA, for instance, could get small enough where you could potentially find an RNA bigger than that. But for the most part, DNA is huge and RNA is small, okay? So DNA contains many units of information. Chromosomes have many, many, many genes in there. And each one gets transcribed and we make just a little segment of RNA from that. So RNA contains individual units of information where DNA contains many. DNA's job is to store genetic information and act as a repository for transcription so that we can later on transcribe and translate that information into some type of protein. Okay, so I like to think of DNA as the hard copy. Okay, that's your permanent storage molecule, whereas RNA is used to express information. So RNA typically doesn't hang around a long time. DNA, in theory, should sort of last forever. Okay? Eventually, when you die, it will get broken down. But inside your cells, the DNA is, in theory, supposed to be permanent. Okay? Whereas RNA gets turned over. Okay? It may last a few minutes or a few hours, but usually at most, it's not going to last longer than a few days before it gets turned over. Okay? So it's a transient expression molecule. So together, the three types of RNA we're going to talk about are three major types. There are other types of RNA, but we're just going to talk about the three major types. Act with other protein molecules to allow the information in DNA to be translated into protein. That's what we're going to spend a lot of the rest of this week talking about. The first of the three major classes of RNA is our messenger RNAs. Okay? They're very heterogeneous in size. So the other two major types of RNAs we're going to talk about are very distinct sizes. Messenger RNAs are all over the map. And it really depends on how big the protein is that's going to be translated from that messenger RNA. Okay, that's what messenger RNA contains, a region that's going to be translated into a protein. Okay, it contains the code in there that's going to be translated into a protein. So depending on how big that protein is, that's going to change the size of that messenger RNA. So there's quite a variation in messenger RNAs from, maybe a few hundred nucleotides up into getting in the 10,000 range, and possibly even bigger, depending on the size of the protein. There are some special structures you will see on eukaryotic messenger RNAs. Okay? Now, all messenger RNAs, we'll start with this bottom part. All messenger RNAs, there's an untranslated region on both sides, and then there's a coding region in the middle. The coding region actually encodes the protein. And those untranslated regions are actually important for translation, which I will get to later on this week, and I'll show you what those untranslated regions do. They absolutely have to be there, but they're not translated themselves. However, eukaryotic messenger RNAs have some additional special structures that prokaryotic messenger RNAs do not have. Okay, so at the 5' terminus of most eukaryotic messenger RNAs, we're going to have a methyl 7 guanine cap, or we just call it the cap structure. Okay, it's going to be important for translation, as I'm going to show you. Okay, so it plays an important role in translation. Also plays an important role in stabilizing the RNA. 3' terminus on a Eukaryotic messenger RNA has a poly A tail. Just got a bunch of A's at the end. This is a post-transcriptional modification. Actually, both of these are they're added after the gene is transcribed. Okay, and that poly A tail also has some important functions. It does serve to stabilize it, does enhance translation, and it also, in this case, actually serves as a signal to exit the nucleus. And so prokaryotes don't have those special terminal structures on the messenger RNA molecules. Okay, the next major class of RNAs, we have our messenger RNAs, now we have our ribosomal RNAs. So both the ribosomal RNAs and the transfer RNAs are distinct sizes, okay, unlike messenger RNAs. Okay, so this figure has a little bit more detail. I liked the picture, that's why I included it. This one didn't come out of your textbook. This came out of molecular biology of the cell, but I happen to like the picture, so I used this one. But what I want you, so that help you visualize it. Well, what I expect you to know is what's written right here. Okay. So they give, for example, they're giving you the size of the ribosomal RNA. At this point, I'm not going to ask you to remember the size of the ribosomal RNAs themselves, although I do want you to learn the size of the ribosomal subunits. Okay? There's a reason for that, because you're going to start talking about, as you go through med school, antibiotics. They're going to talk about ribosomal subunits. The quicker you learn it, the easier your life will be. So get them straight in your head now. But what I do want you to know about ribosomal RNA, not the specific sizes, but I do want you to know that we have 3 distinct ribosomal RNAs in prokaryotes and 4 in eukaryotes. You don't have to know the sizes, but you should know that there's 3 in prokaryotes, 4 in eukaryotes. They combine with proteins, and actually a lot of proteins, as you can see here, 49, 33, a bunch of proteins. So ribosomes are really very complex machines. Contain both multiple RNA units and a whole bunch of different proteins. Very complex. But they combine with these proteins to form ribosomes. And ribosomes can actually be separated into a small and large subunit. Okay? And they're distinct sizes themselves. Now the sizes, you'll see these 50S, 30S, 70S. You'll see these S's always given. Sometimes that can confuse people if it's never been explained what the S actually means. This is a Svedberg, which is a unit of sedimentation. So they define the size by actually taking these things and centrifuging them. That means when you actually look at a whole ribosome versus the subunits, they're not additive. Like, for example, you'd say, okay, we have a 60S large subunit in eukaryotes and a 40S small subunit. Well, if you add them together, it should be 100, right? But it's not. It's 80. Well, why is that? It's because we're looking at sedimentation. Okay, if they were separate, if you just took 60 and 40 and put them together and they went through like that, it would add up to 100, but they kind of overlap. So they're going to sediment a little bit faster than 100, but they're going to sediment slower than either one alone. Okay, so they don't add up. That's why they don't add up. So unfortunately, you just have to commit those to memory. And unfortunately, with the size of ribosomes, it is one of those things that is, you're going to see this over and over throughout med school, in particular when you get into microbiology and start talking about antibiotics. So I'm just going to ask you to go ahead and learn them now, so make your life easier. So eukaryotes, the whole ribosome itself is an 80S subunit. The large subunit is 60, the small subunit is 40. And then prokaryotes, we have our 70, the whole thing, our 50 and 30S. 50, the large subunit, 30, the small subunits. And the ribosomes at various times exist as possibly a whole ribosome or the individual subunits. And we'll see that when we talk about translation. Okay? So basically, all the information that I expect you to know is written down here. The picture is to help you visualize. There's a little bit of extra detail there we're not going to talk about. Okay, our third major class of RNA Once again, there's more RNA molecules that we're not going to talk about. There's 3 big ones. We saw messenger RNA, ribosomal RNA. Now we have our transfer RNA. Transfer RNAs are approximately 80 nucleotides, so they're the smallest. Some people remember that because T sounds like T, tiny RNAs. So transfer RNAs are the smallest. They're very unique in structure, though. They can be covalently linked to a specific amino acid when we charge them, which we're going to see. Okay, so they're actually going to be, this is actually a nucleic acid, amino acid hybrid. Okay, so this is going to be the link where we get, we go from a nucleic acid molecule to a protein. Okay. So they're covalently linked to a specific amino acid. That means a particular transfer RNA, and there's going to be a whole bunch of different ones. It's going to be specific to a particular amino acid. And that amino acid will be added on by a separate enzyme. So you have to have at least one specific transfer RNA, and in many cases there's more than one for each of the 20 amino acids. And we'll talk a little bit about the genetic code, I think, tomorrow. Also has a distinctive 3' terminus, the CCA terminus. It's actually added on there post-transcriptionally, but all mature transfer RNAs have that CCA terminus. That's where we're actually going to attach the amino acid. They have a cloverleaf-like structure. That's what we're showing here with these. If you loosely use your imagination, it kind of looks like a 3-leafed clover right there. So we actually have some base pairing back on itself. If you heat this up, it's actually a single-stranded molecule, but it's got regions where it folds back on itself. So right in these regions, it's not giving you the exact sequence, so you have to trust me that it's actually complementary right there. Okay? And though that folding back on itself, giving it this 3-dimensional structure, is actually vitally important for a transfer RNA to work correctly. You also have unusual bases. So you'll see these Greek characters in there. You also see there's a T there. You don't usually see T in RNA. Transfer RNA is the exception. So there's a bunch of methylation reactions that will happen. What happens if you methylate U? If you look back at the picture and you add a methyl group to U, you actually make a T. So it's actually a ribonucleotide in there. It's still it's still got the 5-carbon ribose in it, but the base has been modified from a U to a T. So some of the uracils in a transfer RNA will actually be modified to a T. We also have this anticodon loop, which is right down here. It's actually, so the loop is actually a little bit bigger, but the anticodon itself is going to be these 3 nucleotides. And as we're going to see, those 3 nucleotides are actually going to base pair with the messenger RNA in the process of translation. Okay, so that's our transfer RNA. It's got some unique structures. And so once again, in the textbook, there was a couple of figures near the end that I'm not going to sit here and walk through with you, but I included them there because sometimes they're useful as kind of a summary to summarize at least some of what was in the PowerPoint. So use those as you guys see fit. I really like the RNA one because it's pretty much got everything in there. The DNA one is not bad either. Okay? And so that's all for this lecture, and I'll see you in 10 minutes.